This video describes the process of the cryopreserved suspension hepatocyte assay commonly used to determine in vitro clearance. What is hepatocyte clearance? Metabolism can have an important effect on the toxicity and therapeutic range of a drug. Drug metabolism occurs in many tissues such as the kidneys, lungs, skin, gut wall, and blood. The liver is the principal site of metabolism, with liver hepatocytes and microsomes being used as an effective way to determine in vitro drug clearance. Hepatocyte clearance. Unlike microsomes, hepatocytes are full liver cells which contain the whole package of phase 1 and phase 2 drug metabolizing enzymes. As a result, both phase 1 and phase 2 metabolism can be tested and determine clearance that reflects cellular metabolism. The use of in vitro metabolism studies to determine hepatocyte clearance alongside other ADME parameters such as plasma protein binding are a good resource for the prediction of in vivo half-life and hepatic clearance of a drug. Various species of hepatocytes can be used to determine and understand interspecies differences, which is fundamental for carrying out in vivo dose predictions. For this experiment, you will need cryopreserved hepatocytes, CHARM, which is cryopreserved hepatocyte recovery medium, Williams medium E with no phenol red, primary hepatocyte maintenance supplement CM4000, trypan blue solution, dimethyl sulfoxide or methanol, acetonitrile plus a suitable internal standard. Any suitable internal standard can be used, for example donepezil. This must be used at a concentration that does not saturate the MS. Internal standards are used to correct variability during LCMS analysis. These must be similar to the analyte, but also distinguishable when analysed. You will also need 8218 acetonitrile water and positive controls such as verapamil, 7-ethoxycoumarin, and thalazine. Controls are required to ensure the assay is working correctly and the hepatocytes are alive and metabolizing as expected. The controls chosen should test each of the different metabolic pathways. For this experiment, you will also need hemocytometers, 48-well plates for suspension cells, a water bath, a thermometer, pipettes which are either single or multi-channel, 96-well deep well 2 ml blocks, 96-well 300 microliter V bottom plates, 0.5 ml microtubes, 50 ml centrifuge tubes, a timer, DMSO resistant plate seals, a centrifuge, a microscope, an LCMS instrument which is liquid chromatography with tandem mass spectrometry, and an incubator with 5% CO2 and a plate shaker. Alternatives can be used where necessary. For example, alternatives can be used in place of the 96-well deep well 2 ml blocks and 96-well 300 microliter V bottom plates. You could use MS vials or microtubes as alternatives. If you are using alternatives, please ensure that the final carrier with your samples for MS analysis is suitable for use on the instrument. For example, use MS vials or 2 ml deep well blocks with a pierceable seal. Assay preparation. Firstly, remove the WME and supplement pack CM4000 from the fridge. Add the whole supplement pack to the 500 ml bottle of WME. The adding of the CM4000 supplement is only carried out when a new bottle of WME is opened. Once added, the supplemented media can be stored at 2 to 8 degrees Celsius for up to 1 month and used as needed. Bubble the WME with carbogen, which is 5% CO2, for at least 30 minutes prior to use. While the WME is bubbling, take out 3 48-well plates. Label 1 cells, 1 WME and 1 no-cell control. You can also heat up your water bath at 37 degrees Celsius and create your termination blocks by adding 100 microliters of acetonitrile plus a suitable internal standard to a whole 96-well deep well 2 ml block. Create the number of termination blocks depending on your number of compounds. There are 8 compounds per termination block. For example, 16 compounds equals 2 termination blocks. Next, warm up your WME and charm in the incubator to 37 degrees Celsius for around 30 minutes prior to initiating your experiment. Ensure you check the temperature with a thermometer before starting. Compound preparation and cell thawing. Firstly, gather your compounds and controls and prepare them to 100 micromolar in DMSO. It is important that controls such as 7-HC, which are metabolites, need to be prepared tenfold higher. These should be prepared to 1 millimolar. Next, in the 48-well plate labelled WME, prepare 1 ml of a 1 micromolar working solution for compounds and controls in supplemented WME. However, here, as compounds such as 7-HC need to be at a concentration tenfold higher, they will be prepared as 1 ml of a 10 micromolar solution. Cover the plate with lid and incubate at 37 degrees Celsius, 5% CO2 until use. Next, retrieve 1 vial of cryopreserved hepatocytes from the liquid nitrogen store and transport on dry ice. Thaw according to the manufacturer's instructions, for example, in a 37-degree water bath. Once thawed, tip the full vial of hepatocytes into the preheated charm. Mix gently and centrifuge according to the manufacturer's instructions. Different species are centrifuged at different speeds. Check the manufacturer's instructions to ensure the hepatocytes are centrifuged at the correct speed. Cell preparation and counting. Once centrifuged, tip the supernatant from the charm in one slow tipping motion, leaving the hepatocyte pellet. Gently resuspend the pellet in 1 ml of supplemented WME. To a microtube, add 80 microliters of supplemented WME, 10 microliters of trypan blue, and 10 microliters of resuspended cells, and mix. Add to both sides of a hemocytometer. Refer to the manufacturer's instructions for volume. Count the number of live and dead cells in both sides of the hemocytometer using a microscope. To count your cells using the hemocytometer, please use the method as follows. Focus until you can see squares that look like this on either side of the hemocytometer. There should be 4 quadrants of 16 squares on each side. Using a hand counter, count the number of live cells. These are cells which have not been stained by the trypan blue. Also count the number of dead cells. These are cells which are stained by trypan blue. Trypan blue is a stain used to quantify live cells by staining dead cells. Trypan blue can only penetrate cells with damaged cell membranes and is therefore only able to penetrate dead cells. Please ensure that the same guidelines are used for counting each box of 16 squares, i.e., cells counted are fully within the squares. Count the cells in the 16 squares in each of the 4 quadrants on both sides of the hemocytometer, and use this to calculate cell viability. Calculation of cell viability. Note the number of dead and alive cells counted in each quadrant and calculate the total number of cells in each. Calculate the percent viability for each quadrant. Viability equals 100 divided by the total number of cells times a live number of cells. Calculate the averages for the number of cells and percent viability. Now, you can calculate the volume required to dilute cells to 0.5 million cells per ml. Dilution of cells to 0.5 million cells per ml. To calculate the volume required to dilute cells to 0.5 million cells per ml, please refer to the cell count previously determined. Firstly, multiply the average number of live cells by the dilution factor to give cells per ml. The dilution factor is the factor which we diluted the resuspended cells before we counted them. For example, we added 10 microliters of resuspended cells to 90 microliters of trypan blue WME mix, therefore giving a dilution factor of 10. For example, in this case, the number of live cells is 59, times the dilution factor, which is 10. This will therefore give 590 cells per mil. Multiply by the hemocytometer factor and convert to million cells per mil. The hemocytometer factor will be stated in the manufacturer's instructions. For example, 590 times the hemocytometer factor, which in this case is 10,000. This therefore gives 5.9 million cells per mil. Next, calculate the volume of WME needed to dilute cells to 0.5 million cells per mil. To do this, please follow the following equation. 5.9 million cells per mil divided by 0.5 million cells per mil minus the volume of WME the cell mix is resuspended in, which is 1 mil. This therefore gives 10.8 mils. Therefore, dilute the resuspended cells in 10.8 mils of supplemented WME to give 0.5 million cells per mil. Cell dilution and plating. Dilute resuspended cells to 0.5 million cells per mil using the calculation on the previous slide. For example, dilute resuspended cells in 10.8 mils of supplemented WME. Next, in the 48 well plate labelled cells, add 200 microliters of 0.5 million cells per mil to one well for each compound and control. A quick tip is to aliquot the cells in the same layout as the WME plate previously created. This will make sampling easier. In addition, aliquot an extra well for a cell viability check at the end of the experiment. Next, incubate the plate for up to 10 minutes at 37 degrees Celsius, 5% CO2 and gentle agitation, approximately 90 rpm, prior to use. No cell control initiation and sampling. Firstly, label up each termination block with each of the time points, which are 0369, 1530, 4560, 90, 120 minutes. Column 1 is T0, column 2 is T3, and so forth. The last two columns in each termination block are used for the no cell control. No cell control samples are taken at 0 and 120 minutes. Please label these accordingly. Next, from the 48 well WME plate, remove 200 microliters of the 1 micromolar compound and controls in column A into column A of the 48 well no cell control plate already containing WME. Agitate this plate slightly. Immediately sample 20 microliters into the appropriate column in the appropriate termination block. This is no cell control T0. For example, column A equals termination block 1. Repeat these steps for column B and sample into the appropriate termination block. Column B equals termination block 2. Once all columns have been sampled, place the no cell control and WME plates back into the incubator. Experiment initiation and sampling. From the 48 well WME plate, remove 200 microliters of the 1 micromolar compounds and controls in column A into column A of the 48 well cell plate containing 0.5 million cells per mil. Agitate this plate slightly. The final working solution concentration is 0.5 micromolar and the final cell concentration is 0.25 million cells per mil. Immediately sample 20 microliters into the appropriate column in the appropriate termination block. This is T0. For example, column A goes into termination block 1. Ensure that after sampling you start your timer. Repeat these steps for column B and sample into the appropriate termination block. Column B goes into termination block 2. From the cell plate, sample 20 microliters of the 0.5 micromolar working solution at 369, 15, 30, 45, 60, 90 and 120 minutes for each of the columns in the relevant termination block. It is important that you ensure that you stagger your sampling so that each column is accurately sampled at the correct time. No cell control T120 sampling. At the end of the experiment, sample 20 microliters of the no cell control working solution into the 120-minute column in the appropriate termination block. For example, if you are sampling from the no cell control plate, column A, dispense into termination block 1. Repeat these steps for column B and sample into the appropriate termination block. Column B goes into termination block 2. T120 viability check. Carry out a viability check at the end of the experiment to ensure cells are viable. To a microtube add 90 microliters of cell mix from the extra well in the cells plate and 10 microliters of trypan blue. Mix this gently. Add mix to both sides of the hemocytometer and count cells in the same way as previously carried out. You can now calculate the T120 viability for the end of the experiment. Calculate using the same formula as used for the viability check at the beginning. 8218 addition. Add 100 microliters of 8218 acetonitrile water to each of the termination blocks. Centrifuge blocks at 3750 rpm for 10 minutes. Finally, seal your samples with an appropriate seal and they are now ready to be analysed by LCMS. Data analysis, creating an exponential decay curve. When your samples have been run, you will receive raw mass spec data. From your raw data, use the response at each time point to create an exponential decay curve of response against time in minutes. This can be done using a graphical package, for example XLFIT or GraphIt. Response is the analyte and internal standard response ratio. Response is used to reduce analytical variability of results. Once plotted, your graph should look as follows. Data analysis, the calculation of clearance in mils per min per gram liver. Clearance in mils per min per gram liver equals K divided by V times the hepatocyte scaling factor, where K is rate per minute and V is million cells per mil. K is the decay rate derived from the exponential decay equation. The exponential decay equation is y equals ABX, where A is the initial amount, y is the final amount, x is the number of time intervals passed, B is the decay factor, where B equals 1 minus R, where R equals rate, which is also written as K. V is the final concentration of cells used in the experiment in million cells per mil. In our case, this is 0.25 million cells per mil. Hepatocyte scaling factor is a constant which changes depending on which species is used. Mouse, rat and human is 120 million cells per gram liver, and dog is 240 million cells per gram liver. Data analysis, an example calculation of clearance in mils per min per gram liver. Below, we have our equation and we have our key. Our example equation uses mouse hepatocytes. K, which was calculated from our exponential decay curve using the data previously shown, was given as 0.044 per min. V is 0.25 million cells per mil, and the hepatocyte scaling factor, as our species is mouse, is 120 million cells per gram liver. Clearance in mil per min per gram liver is calculated as 0.044 divided by 0.25 times 120. Therefore, clearance equals 21 mils per min per gram liver. Data analysis, example calculation of half-life. Half-life, also known as T half, equals ln2 divided by k, where k is the rate per minute and ln2 is the natural log of 2, calculated as 0.693. Half-life is the time taken for half of a drug to be broken down/metabolized by the hepatocytes. For example, K equals 0.044 per minute, which we've calculated previously from our exponential decay curve, and ln2 is the natural log of 2, which is 0.693. This is a constant. Therefore, T half in minutes equals 0.693 divided by 0.044, therefore giving a half-life of 16 minutes. So what to do with hepatocyte clearance results? You can utilise the data as an early stage screening tool to triage compound progression. You can correct for other ADME parameters such as plasma protein binding and scale in vitro clearance data to predict in vivo hepatic clearance. You can use the data to compare and investigate interspecies differences in clearance. Or the hepatocyte method can be adapted to carry out other techniques such as metabolite identification. Advantages and disadvantages of suspension hepatocytes. Advantages. Hepatocytes are full liver cells which contain the whole package of phase 1 and phase 2 drug metabolizing enzymes. Both phase 1 and phase 2 metabolism can be tested and clearance determined that reflects cellular metabolism. Hepatocytes provide an effective in vitro model for predicting in vivo metabolism. Minimal amount of compound is needed, and additional cofactors are not needed for enzyme activity. Disadvantages. Hepatocytes are expensive compared to other methods of determining metabolism, for example microsomes. The assay is not high throughput, and suspension hepatocyte assays can only be taken out to around 2 hours before cell viability and metabolic competency is reduced, therefore resulting in suspension hepatocytes not being optimal for determining an accurate clearance value for low turnover compounds.